Hello my dear students, this is Dr. 𝗲𝗵𝗵𝗺, your biochemistry faculty. Well, in this video we are going to discuss about a very interesting topic. We are going to discuss epigenetics and genomic imprinting, right? So let's start up with the discussion. Epigenetics and genomic imprinting. Now, these two are very important terms, widely used nowadays. Okay, so first of all, what do you mean, what do you understand by genetics? Genetics is simply the genetic organization, genetic information which is present in a individual or a human being. And if we talk about the genome, it is the entire hereditary constitution of an individual. So if we have to define genome, genome is simply the entire hereditary constitution of an individual, right? Constitution of an individual. And whenever we talk about the genome, we know that haploid is there, diploid is there. And if we talk about the total number of chromosomes we know that 23 pairs are there. And out of this 23 pairs, one pair is of the sex chromosomes and rest represent the autosomes. And if we talk about the sex chromosomes, we know that we are having heterogametic males, right, which have a XY genotype, and we are having homogametic females which have a XX genotype, right? Now the question is, this is just the genome, so it can be haploid, it can be diploid. Now what is this epigenetics? What is its significance and what is this genomic imprinting? If we talk about genome, I have discussed, imprinting is, it's like silencing one particular gene. Imprinting means कि जैसे suppose if two are expressing themselves, if one is silenced, the other will automatically express, right? So this process is known as genomic imprinting, okay, which is very, it's not a very frequent process. Less than 1% of cases may it has been observed, but yes, it is a very important process, okay. And epigenetics is simply something which is overpowering that. So it's like, just the example of epigenetics I would say is methylation. DNA methylation is a very important process of epigenetics. So what happens is, these are two different terminologies. If we talk about epigenetics, so here we can simply say epi mane upon a gene, you can say like that. So what happens in epigenetics is that DNA code never changes. Just keep this thing in mind. There is no change in the DNA code. It's just a modification. So there is no change in DNA code, but it will be modified. So we can say that DNA can be modified. And the most important point about this is, this process is reversible, okay. So this process is reversible. Now there are two methods in epigenetics. First is, we can say DNA methylation. And second is post-translational modification of histones. Post-translational modification of histones. Now, first of all, we will take the methylation first, and then after that we will take the PTM of histones. Now, if we talk about DNA methylation, that means DNA is undergoing attachment of a methyl group. So, where this methyl group is going and attaching itself. So, what happens is this methylation process will only occur on the 𝗵𝗵𝗵 islands, or it will occur only on those sequences which are having CG on one strand. It is not like this कि it can C can be on one strand, guanine can be of another strand. No. DNA methylation के लिए the mandate has to be that it should be rich in CpG islands. What do you mean by CpG islands? Means cytosine and guanine should be placed at one strand. Okay, so DNA methylation के लिए the most important thing is that we can say that occurs only on CpG islands. What does it signify? It signifies that CG base pairs should be more. Should be more. Very important, right? And should be placed at one strand. Should be placed on one strand. Let's say, for example, if I say this is one strand, this is another strand, right? So, if we talk about this, cytosine is here, guanine is here. And if we talk about there are two strands, cytosine and guanine. So, here no methylation will occur, but this will become a site of methylation. So, methylation will occur here. And simple, if methyl group is attached, obviously there will be a modification in DNA. That DNA will be known as a methylated DNA. Once the function is over, it can be easily removed also. And this DNA methylation can be detected by sodium bisulfite method. So, this is detected by sodium bisulfite method. So, again, if we talk about the significance of this, we are simply adding a methyl group. Sometimes it can be harmful, sometimes it can be useful also. So, it is also known as a epigenetic modification, right? So, in the epigenetic modification, DNA methylation plays a very, very important role, which occurs only on the CpG island, which means that the strand which is having cytosine and guanosine residues. Then second is a very important method, which is post-translational modification of histones. Now, if we talk about histones, first of all we should know that what are these histones. So, these histones are the proteins rich in basic amino acids. And what are these basic amino acids? Lysine and arginine. And if we talk about the structure of DNA or the organization of DNA on a chromosome, because we know DNA is the heredity material, isn't it? And DNA which is present on the chromosomes is transmitted from one generation to another generation. Okay, so what happens here is these histones are actually 4 in number, which are present in a doublet. So, they are of 4 types: H2A, H2B, H3 and H4, and they are present in a doublet or double. right? So they will become, they are going to form an octamer. And H1 is also present, right? H1, which is also known as spacer or a linker DNA. We can call it as a linker or a spacer DNA, right? Now if we talk about this, so we can say that H2A, H2B, H3, H4, since they are present as an octamer 5, 6, 7 and 8. So this DNA is intertwined like this, okay. So it is known as a nucleoprotein. So what is a nucleoprotein? Nucleus means DNA which is combined with histones. And we call this structure as nucleosome. Nucleosome, okay. Now the question is that how this nucleosome is arranged on a chromosome. So if we talk about the organization, I can simply say that this is the chromosome like this, right? And this is the thread-like structure. It is known as chromonimeta. Okay, so on this particular, this is the centromere, alright, and these are the sister chromatids. So what happens is this structure, if you are seeing this nucleosome, it is present here. So like this, like this, like this, like this. And there is one more chotu sa, I would say, this is known as linker DNA. Okay, so I can call this as a nucleosome, this one, this structure. This is a nucleosome which is DNA plus histones, and this red one is linker DNA or H1, or you can say linker DNA or spacer DNA. So what happens here is this is representing beads, looking like beads as if necklace is there. So this is known as a beads on a string appearance. And depending upon this, we can simply say that because chromosomes are the ones which are bearing the chromatin or the fibers. So depending upon this, we obviously generally stain that just to check where the staining area is there, and when we can actually well define the structure also of this. So what happens here is that if we put a stain, the lightly stained areas are known as euchromatin. The dark stained areas are known as heterochromatin because they will take more. Okay, so on the basis of this, so we can say that there are two types of chromatin fibers. And these chromatin fibers can be euchromatin and heterochromatin. And if we talk about the basic difference between them, euchromatin and heterochromatin mein the most important difference is they are less stained. They are darkly stained or dark stained. They are active transcriptionally, or transcription is active here. Is active. And here transcription is inactive. And the third difference is it has, regarding the chromatin, it has permissive chromatin, and it has repressive chromatin. repressive chromatin, right? So this is the basic difference between these. Now here we are discussing about histones. So what is this epigenetic modification, or you can say what is this post-translational modification? Now just understand one thing that we know. If we talk about the central dogma of information, we know that this DNA is converted into RNA, okay, and this RNA is converted into proteins, or it undergoes translation. So DNA to RNA is transcription, right? RNA to proteins is translation, and DNA to DNA is replication. Now the question is, we always say that histones can undergo, या histones can regulate the genes, because histones are the ones which are gene regulators. They are ready to express the gene. Now the question is how this gene regulation is carried out. There are so many different mechanisms in the support of this, but here since we are talking about only histones, so we will be targeting only about that. And यहाँ पे I can say RNA can be converted into DNA. Now the question is, DNA is already wrapped around histones, and it is so compactly arranged that nucleosome, till the time DNA is not released from the vicinity of the histones, it cannot perform any function. So DNA will be lying as it is, like that. That is why there are different phases of cell cycle also. Okay, so which process occurs in which phase of cell cycle will determine the fate of DNA also, because the replication will occur only in the S phase. So now the question is that how this DNA will come from the vicinity of histones so that they can undergo transcription. That is what is this post-translational modification all about. So जैसे DNA methylation is there, just parallel to this one more process is there, we call it as acetylation or deacetylation, right? So what happens is acetylation or deacetylation. Now what is happening is, we have this type of structure. I would just represent it as a nucleosome like this. This is a nucleosome where lysine is present as the principal amino acid, because lysine and arginine are the two basic amino acids present in histones. So what will happen is acetylation will occur. We can call it as acylation also. Basically acetylation means CH3 C double bond O, like this. So what happens is, when it undergoes acetylation, lysine will undergo this process. So lysine will be busy undergoing acetylation, DNA will be released like that. So the name of the enzyme here will be histone acyl or acetyl transferase, or commonly it is known as HA. So what will happen is, lysine will undergo the process. So when lysine will undergo the process, that means DNA will be released. DNA is released. Now when DNA is released, that means it is free. So it is going to transcribe RNA. Right? So that is why we say कि histones play a very, very important role in regulation of the gene expression. So lysine will be required here, transcription हो गया, then RNA has to undergo translation, transcription is over. So what will happen is, the cell will get instructions and immediately another enzyme will come into function, which is histone deacetylation, or you can say HDAT, which stands for histone acetyltransferase. Now what is its function? It is going to remove acetyl group from lysine. Now it has removed the group from lysine. Lysine is now occupied. So what will happen is lysine goes inside and packs DNA again, and DNA gets packed. Okay, so it's like I would say that acetylation and deacetylation are the two very important kind of processes which are actually a part of epigenetic modification. So what we are doing is we are simply acetylating it. So acetylation say if because lysine is there, so lysine will undergo the process and lysine will be released because acetyl group is already attacking lysine. So when lysine attacks it, DNA is free. When DNA is free, automatically it will help in the formation of RNA. Once transcription is done, because the cell is getting that signal that this is the right time to synthesize RNA, this is the time not to synthesize RNA. So immediately deacetylation will be there, and this enzyme will take acetyl group from lysine. So if lysine goes inside, DNA gets packed, so no transcription is there. So this is again a very important process, and we call it as post-translational modification also. And the most important thing is I can say that this is also reversible. Why this is reversible? Because acetylation and deacetylation is going side by side, right? So I can say that since it is reversible, which means that acetylation and deacetylation are just changing the conformation of lysine as per the need of the body. That is why in the starting only I told you there will be no change in anything. DNA code is not changing. No sequence is changing. It is simply, I would say, modifying that. Right? So acetylation and deacetylation are the two very important processes which can help in activating or inhibiting transcription. So if I talk about acetylation, I can say it activates transcription. And if I talk about deacetylation, it inhibits transcription. Very important, right? So epigenetic modifications, there are two. One is DNA methylation. DNA methylation may always remember one thing, that methylation of DNA is always carried out on the CpG island. CpG island means that cytosine and guanine residues are, but they should be present on one strand. I told you this is not permissible, and it can be detected by sodium bisulfite method. And second process is the post-translational modification of histones, and histones mein the process is acetylation and deacetylation, depending upon it is HAT or HDAT enzyme, depending upon whether it is activating, whether it is inhibiting. And if we have to detect this post-translational modification, it is detected by chromosome or chromatin immunoprecipitation method. Chromatin immunoprecipitation method. Very important. Mnemonic can be 𝗲𝗵. So you can just remember 𝗲𝗵. CH for chromatin and IP for immunoprecipitation. Okay. So this is epigenetic modification. Now second term parallel to this is genomic imprinting. Now what is that genomic imprinting? Genomic imprinting. Imprinting is another word for silencing. Now, silencing means any change in the conformation of that. So, what happens is, if this genome, first of all, you can say that imprinting is silencing of one allele. So, the other can be expressed. The other will express itself. Express itself. This is again very, very important. So, genomic imprinting is basically simple. You can say silencing of one allele, the other is going to express itself. And normally it is like less than 1% is seen. So, if we talk about this, there can be two types of imprinting. We can say one can be paternal imprinting. And second can be maternal imprinting. Right. Now, what is the basic difference between maternal imprinting and paternal imprinting? If we talk about the paternal one, that means we can say under paternal, which allele is suppressed? Paternal is suppressed. So, paternal allele is suppressed or silenced. Silence is the or imprinted, whatever you can say. So, if paternal allele is suppressed, who is going to work? Maternal will work. So, maternal allele will work or will express. Maternal imprinting में what will happen is कि maternal allele is silenced. Allele is silenced and paternal allele will express. Right. So, this is simple, we can say the genomic imprinting. Now the question is that there are two syndromes which are associated to paternal and maternal imprinting. So, what happens is that there is one Prader-Willi syndrome, Prader-Willi's syndrome and Angelman's syndrome. So, what happens in Prader-Willi syndrome? Okay, so in normal individuals, if we see, it is going to affect the chromosome 15, right, expressed by say ABC gene and segment is Q11 to Q13. Now, what is happening here is Prader-Willi syndrome is an example of uniparental disomy. Example of uniparental disomy. Now, what happens here is, in Prader-Willi syndrome, the maternal allele is suppressed. Yeah, it is silenced. So, the paternal will work. But in PWS, what happens is both the alleles stop working. Okay, I'll just repeat it again. In Prader-Willi syndrome, generally maternal is the one which is suppressed. So, what will happen? The paternal is still working, but in PWS, both will stop working. Okay. So both will stop working. Now, what will be the consequences in Prader-Willi syndrome? For this, there is a mnemonic. It's a very interesting mnemonic because P for Prader-Willi's, or the mnemonic is say 𝗵𝗵, P-A-P-A. So what happens here is P will stand for Prader-Willi, right? A will stand for appetite. The child who is born with Prader-Willi syndrome, he will have a maximum appetite, means he will be all the time hungry, or whatever he is eating, like it's like, it will lead to obesity. So he will be like all the time eating, so we call it as polyphagia. So much eating will be there, it will lead to pediatric obesity. Very important, right? So we can say that Prader-Willi syndrome में more appetite will be there. So जब more appetite is there, so it's like he will have very chubby cheeks. So we call this baby as a floppy infant baby also. Floppy infant baby. Baby with chubby cheeks. Right. So much obesity is there, so much of polyphagia is there. So it will lead to insufficiency of the androgens. So there will be androgens insufficiency. Sufficiency. Very important. So that is why we say it's like PAPA, Prader-Willi syndrome में there is appetite. So polyphagia is there. So much of eating by the child is there, which is leading to the insufficiency of the androgens. Okay, and it might also lead to hypogonadism. Right? Then this is floppy infant syndrome also. Then second is parallel to this, which is Angelman's syndrome. Now, in Angelman syndrome, what happens is, this is also an example of uniparental disomy. Parental disomy. So here what happens is, here paternal is not working. Okay, but in Angelman syndrome, both will not work. So obviously it will lead to the diseases. One more character here is there in Prader-Willi's, hypotonia will be there, because floppy infant का मतलब ही क्या है कि the tonicity in the muscles will be lost. So I can also write here, but I just wanted कि this mnemonic should be learnt because it is easier to remember this Prader-Willi syndrome like this. Otherwise, hypotonia is also there. Okay, so Angelman syndrome, again uniparental disomy is there. So what happens here is, the child who is suffering from Angelman syndrome will all the time be like, a puppet. So it is also known as 𝗵𝗵𝗵𝗵𝗵 syndrome. 𝗵𝗵𝗵𝗵𝗵 syndrome. And here what happens is, there is abnormality in the XYZ gene of again 15th chromosome, segment is Q11 to Q13. Same. But both are different. In one paternal is the one, here in maternal is the one, because that is what I am telling you. In Prader-Willi's, maternal is imprinted. Okay, here in Angelman syndrome, it's opposite. It is maternal is imprinted. Okay. And what happens here? Here also you can say mama. M-A-M-A is the mnemonic to remember Angelman syndrome, right? Now, if we talk about this, I told you ki all the time that, the child who is born will be laughing. So we can say that the child, A say it is ataxis. or there will be lack of coordination, or he will be jumping like puppet. Jumping like a puppet. M say you can say maternal. Right. M say another M stands for mental retardation. And A will stand for, it's like very awkward laughter. A will stand for awkward laughter. And that is why we say in Angelman syndrome, it will be like, that is why Angelman is also known as 𝗵𝗵𝗵𝗵 syndrome. Okay, so I can write it as, and I have written here also that it is known as 𝗵𝗵𝗵𝗵 syndrome. So this is the basic difference, and I'll just show you the image, what is actually happening here. See, right, Prader Willi, or simple way to remember is, just remember Prader Willi mein paternal deletion is there. So P for Prader, P for paternal. That is why I told you Prader Willi ke liye the mnemonic is 𝗵𝗵𝗵𝗵 or Papa. And if we talk about Angelman syndrome, Angelman mein maternal deletion is there. So it's like Mama. Okay, so Papa, paternal deletion, Prader Willi is Mama, maternal deletion, Angelman syndrome. And if we talk about the features here, you can see hypotonia in infancy is there. Hypotonia means it's like the tonicity of the muscles is lost. Okay, and you can see the abdomen here. Obesity is there. Why? Because the appetite increases. Okay, appetite is increasing too much, and the person tends to eat more and more. There is no tonicity in the muscle. Developmental delay will be there, or you can say short stature will be there. Hyperphagia will be there. Hyperphagia, which will obviously lead to obesity. Then hypogonadism. Hypogonadism will obviously be there because you can see the weight is here. It's like the abdominal weight is there. Okay, so which will lead to insufficiency of the adrenals, right? And it will always lead to, but this will be very mild intellectual disability will be observed in Prader-Willi syndrome. Very, very important. We can call it as PWS also. And if we come over to Angelman syndrome, Mama, so we can say it's like M say you can, yes, M say you can remember microcephaly also. Okay. It depends on you ki how you can, make your mnemonics. A for ataxia, because like a puppet, जैसे puppet का gait होता है, like that rope is there and it's like swinging here and there. Then mental retardation will be there. This is very, very important, and this is the basic difference between, I would say, Prader-Willi's and this syndrome. Severe mental disability will be there. Severe intellectual disability. Okay, and then it will lead to inappropriate laughter. It's like if some abnormal man is laughing somewhere. So that is why, and it will lead to seizures also. Microcephaly will be there. Okay, so these things are a characteristic feature of Angelman's syndrome, and this is how we differentiate also. So it's like 𝗵𝗵𝗵𝗵 syndrome, very important, and Papa, or you can say paternal deletion, it is known as floppy infant syndrome. And both are the cases of uniparental disomy. Right? And this is a very, very important example of genomic imprinting, where one gene is silenced. And the chromosome affected is chromosome 15, or segment is Q11 to Q13. Right? So this is all about, second, I'll just take it up. So we can say that this is all about DNA methylation and genomic imprinting, which is very, very important. Epigenetic modification mein the first thing which you have to learn is the most important epigenetic modification is methylation. And always remember, whenever we talk about DNA methylation, okay, it is always on CPG islands. It is always on CG. It is not going to affect adenine or thymine. It is only going to affect C and G. And it is checked by sodium bisulfite method. You can see here. This is very important. Yes. Okay, and second is simple post-translational modification of histones, where the process is acetylation and deacetylation. Now, the most important point which you have to remember, it is always reversible. It cannot be irreversible. If it is irreversible, then again there won't be any genetic expression, there won't be any genetic modification, everything will come to standstill. So always keep this thing in mind. All these are, or you can say these are nothing but these are the post-translational modifications which are reversible. Whether it is methylation, whether it is acetylation, whether it is phosphorylation, whether it is hydroxylation. This becomes the basis of genetic expression. Okay, so I can quote this also as a kind of modification. But yes, why we call it like this? Because it is like upon. Upon means it is modifying. So modification will be done and then it will come back to normal. Then again modification will be done, then it will come back to normal. So this is a very, very important topic, right? Epigenetics and genomic imprinting. I hope it is clear to all of you. So this is all about this video. Thank you so much.